Now, C3 cycle or the Kelvin cycle was found by or details were studied by Melvin Kelvin, and which uh, concluded that the first carbon compound which was stable is a 3-carbon compound, that is phosphoglyceric acid or phosphoglycerine. Now the scientist also later tried to understand that whether all plants have PGA as their first product uh, due to carbon or carbon dioxide fixation, or is there any other product being formed. So there was a query, or there was a question in scientists' mind, that whether the first carbon compound which is formed by the carbon dioxide fixation is 3-carbon compound. Now several experiments were conducted on different plants, and which led to the discovery of a group of plants where the first stable product of carbon dioxide fixation was not a 3-carbon compound but a 4-carbon compound. So C3 cycle or the Kelvin cycle, when we say the C3 cycle or Kelvin cycle, over here it was found that the first stable compound was a 3-carbon compound, that was phosphoglyceric acid. But the scientists, when later on they studied, they found that there is another class or another group of plants where the carbon dioxide fixation was not giving a product which was 3-carbon atom. Instead, a 4-carbon compound was formed. That was oxaloacetic acid, and therefore this cycle is referred to as C4 cycle. Also, this was done, or the path was tracked by Marshall Davidson Hatch and Charles Roger Slack. So therefore this cycle is also referred to as Hatch-Slack pathway. So here, in a broad sense, if we understand, the plants in which the first stable compound of carbon fixation is a 3-carbon atom are said to be C3 plants, whereas some plants in which the first stable carbon compound formed after carbon dioxide fixation is a 4-carbon atom, then those plants are known as C4 plants, or this is also known as they are said to follow the Hatch-Slack pathway. So thus we can say that assimilation or the fixation of carbon dioxide, these are the two methods for assimilation or fixation of carbon dioxide. This happens in two ways. That is, the first in which this product is PGA, phosphoglyceric acid, and the other one in which the product is a 4-carbon atom compound, that is oxaloacetic acid. Now, you also have to understand that all photosynthetic plants, they follow Kelvin cycle, but there is the C4 plants actually have a primary stage prior to the Kelvin cycle. So what is happening over here? Before the C3 cycle begins in C4 plants, there is a pre-step, or there is a primary stage. The C3 cycle still remains the main biosynthetic cycle in which plants manufacture or produce the glucose. Now, C4 plants have certain modifications, anatomical modifications, and these anatomical modifications are due to their habitat, because they start with the formation for the different kind of a product for after the carbon dioxide fixation, and they always end up in the Kelvin cycle. Now the C4 plants are actually found in dry tropical regions or tropical desert. So they are dry tropical plants found in those areas, and they have different leaf anatomy. leaf anatomy is different. Leaf anatomy is special. What it is? It is first, it is such that they can withstand high temperature, because the temperature sometimes ranges up to 50 degree or more than that. Also, they respond to high light intensities in a different manner. The prevention of water loss. So there is minimum loss of water, and also to avoid photo respiration. So, in a nutshell, if we study, the C4 plants are found in dry tropical areas or tropical desert areas. The leaf anatomy helps to withstand high temperature, will respond differently to high light intensities, help in prevention of loss of water, and avoid photorespiration. So, we'll be studying about the Hadzack pathway. Now, if we compare the anatomy of the C3 plants with the leaf anatomy of C3 plant with the C4 plants. Now, C3 plants, let us understand first. Now, this is the structure. This is the waxy cuticle. Followed by the upper epidermis. So this is the upper epidermis. Over here. Then it is having the palisade cells, which are rich in chloroplast. Elongated cells, cylindrical, followed by the spongy cells. And the vascular bundles are present over here. So we have the spongy cells, and this is the mesophyll area, mesophyll cells. This is the palisade cell, palisade mesophyll cell. This is the vascular bundle. These are the spongy cells. The vascular bundle consists of xylem and phloem. And this is followed by the lower epidermis. This is lower epidermis. And over here, we have the lower epidermis will have more stomata. So these are the stomatal openings. This is the stomata over here, and it is again lined. So this is the structure of C3 plants. Now C3 plant examples are rice, wheat, eucalyptus. These are the few examples. Cotton is there. Potato. These are the examples of C3 plants. Now, this is the leaf anatomy of the C3 plant. Now, let us compare with the leaf anatomy of the C4 plants. C4 plants have a leaf anatomy such as the examples of C4 plants are this is maize is an example. Then we have the sugarcane, sorghum. These are few examples of C4 plants, and these are slightly different in structure. So this is again the upper epidermis. This is the cuticle lining over here. Then followed by the upper epidermis. This is the upper epidermis or the epidermal cells. Now here if you see, this is the vascular bundle, and this vascular bundle is surrounded by a layer of cells over here. These are large cells, and these cells surround the vascular bundle. This is bundle sheath cells. These are bundle sheath cells, and this is the vascular bundle. Apart from that, they have several other structures over here, cells which are present, and they may be one layer or they may be several layers of these cells, and these cells are rich in chloroplasts. So we see the C4 plants have large parenchyma cells. These are parenchyma cells, and they are present around the vascular bundle. Now, these bundle sheath along with the vascular bundle are said to show the Cran's anatomy. This is known as Cran's anatomy because the crans means the wreath, and a wreath is circular, and similarly the bundle sheaths are enclosing the vascular bundle in circle, and therefore it is the name crans is given to it. So the C4 plants are shown or are known to have the Cran's anatomy, and they are characterized, the bundle sheath cells are characterized by having large number of chloroplasts, and they have thick walls. So the bundle sheath cells have, they are rich in chloroplast, Also, they have thick walls. So the thick walls do not allow the exchange of gases, and also there is no intercellular space between these cells, and there's no intercellular spaces. The bundle sheath cells actually, they carry the water from the xylem to the mesophyll cells because they are surrounding the xylem, that is the vascular bundle, xylem and phloem. So they will be conducting the water and other things from the xylem to the mesophyll cells and finally to the intercellular space. Now let us now try and understand the actual pathway, how it is working over here. Now when we study this pathway, we find that unlike the C3 plants where the entire process is taking place within the chloroplast, over here the process is divided into two parts, because as we have mentioned, before the plant enters into the Calvin cycle, first it has the pre-stage. So over here, let us draw the diagram. This is the upper epidermis. Now this over here is the first mesophyll cells, and followed by the bundle sheath cells. So this is the mesophyll cell and this is the bundle sheath cell. Now the carbon dioxide from here diffuses into the mesophyll cells. Now this carbon dioxide reacts with water to form carbonic acid. So this will combine to form carbonic acid, HCO3. Actually, what is happening? If we do it over here for the clarity, water plus carbon dioxide reacts to form H2CO3, that is the carbonic acid. And this is happening in presence of an enzyme, carbonic anhydrase. So this enzyme actually helps in combining water with carbon dioxide, and this H2CO3 later dissociates or breaks up into H++ HCO3 minus. Now this bicarbonate which is formed over here is accepted by phosphoenol pyruvate. So this HCO3 is accepted by phosphoenol pyruvate, or in short it is referred to as PEP. So this is combining with PEP, and here the PEP is a 3-carbon atom compound. This and it forms later this PEP is combining with HCO3 and it forms a compound that is OAA. Now here this is a 3-carbon atom compound. This is carrying 1 carbon. So over here the 4-carbon atom compound is formed, that is OAA, that is oxaloacetic acid. Now the reaction between the PEP, that is the phosphoenol pyruvate, and bicarbonate is where the carboxylation takes place. Here the carboxylation is taking place. An enzyme PEPCO, that is the PEP carboxylase or PEPcase, helps in the addition of carbon dioxide. So what is happening over here? The carboxylation is taking place over here. Carboxylation is taking place over here, and the enzyme which is responsible is PEPCO, that is phosphoenol pyruvate carboxylase or PEPcase. So in C3 cycle we have talked about Rubisco which is taking part, but here it has changed its nature, and thus phosphoenol pyruvase acts as a carboxylase, whether it is high temperature or low temperature. Now after this the oxaloacetic acid changes either into malic acid, that OAA either changes into malic acid, which is again a 4-carbon atom compound, or it can also change into aspartic acid, which is also a 4-carbon atom compound. Now, whether malic acid or aspartic acid, they will diffuse into bundle sheath cells. So what is happening over here? This OAA changes into malic acid. Over here we have taken malic acid, and it is also a 4-carbon atom compound. This malic acid diffuses into the bundle sheath cell. So here it will diffuse into the bundle sheath cell, and malic acid is, which is having how many carbons? 4 carbons. It enters over here. Now this 4-carbon atom compound dissociates into 3-carbon atom compound, and one carbon is given as carbon dioxide. So this malic acid undergoes decarboxylation. So this undergoes decarboxylation over here, produces a 3-carbon compound which is known as pyruvate, and 1 CO2 is released. 1 carbon dioxide is released over here. This 3-carbon compound pyruvate diffuses back into the mesophyll cell. So again it goes back into the mesophyll cell. This is a 3-carbon atom compound. It diffuses into the mesophyll cells. Now this pyruvic acid is without phosphate because there is no phosphate group attached over here. So this pyruvic acid undergoes phosphorylation to form phosphoenol pyruvate, that is PEP. So over here this undergoes again to form phosphoenol pyruvate, and here it forms the PEP, and during phosphorylation ATP is used and it is converted into AMP, monophosphate. ATP gets converted into monophosphate, and the carbon dioxide is taken up by the RUBP in presence of enzyme. So the carbon dioxide which is being released during this decarboxylation is utilized by RUBP, and it is fixation is done by the Calvin cycle. Calvin cycle over here. The process of carboxylation is over here, and since the enzyme is received or is obtained back, this is known as regeneration over here, because we have got back the PEP, which is the first acceptor. So this acts as a the ATP is being used and is converted into monophosphate. Now the bicarbonate is accepted by the phosphoenol pyruvate, and we get that ATP is being used. Here the RUBP is acting as a carboxylase and it will enter into the Calvin cycle. When we talk of the C4 pathway, the plants are shown to have the plant C4 pathway. The mesophyll cells will be taking part, and the Calvin cycle is done in the bundle sheath cells in the case of C4 plants.